All right, so I apologize for those of you who usually come to the live lectures that I couldn't have one at 2 today. Um, yeah, apologies for that. The way that this is going to work is we have 2 PowerPoints to go over. We have the part from Wednesday we didn't finish up, and then we have the new one for today. So I'm going to get through those. And we have questions like normal, but unlike a live lecture where I pause and give people time to do it, since you're watching this on the video, I'm gonna leave it up to you to pause before I solve it. And as always, like I said at the very beginning of the semester, if you're just waiting for me to give you the answer and you're not putting any thought into it, you're not going to learn this stuff. So please, I do really hope you are pausing the videos, you are trying it, you're making a good effort, you're making your brain hurt to try and understand this stuff. And if you hit a wall, then you can see how I did it. Or if you have a solution, then you can see if it matches mine. So let's begin here. And we're going to start by looking at our modifications of sugars. So far, we've only looked at sugars that have OHs on them. However, many modifications happen to sugars inside of our cells. And when those modifications happen, they get special names. So here, let's start in the top left. If we remove one of the hydroxyls, one of the OH groups rather, one of the alcohols, and just replace it with an H, that is called deoxy. And here, we're actually looking at deoxyribose or the base for DNA. So anytime you remove a high OH group, an alcohol, it is deoxy. It does not have to be at the 2 position, and it can happen to multiple positions. If you take your C double bond O and you make it a carboxylic acid, this is called an aldonic acid, and it has to be an aldehyde. It's not in the ketone form, but if you start with the aldehyde and make it a carboxylic acid, this is called an aldonic acid. And the name of the sugar just changes to ic acid. So glucose goes to gluconic acid. Now, compare that with a uronic acid, and that's where you take the very last carbon and you change that alcohol to a carboxylic acid. So that's called a uronic acid. And again, you take the base name. So we're looking at glucose, and you just add uronic acid. So this is glucuronic acid. You have your alditols. And alditols is, instead of changing your aldose and ketose to a carboxylic acid, as we did with aldonic, all you're doing it is changing it to an alcohol group. So we're going from this to this, and that is called the alditol. And one current or common alditol you might know of if you enjoy gum as much as I do is called a xylitol. So sugarless gum, I mean, technically it does have a sugar in it, just not glucose. So here is xylitol. And then up here, our last modification we're going to talk about is if you replace OH group with amino group, those are called amines. And so you take the name of your sugar and you just add amine to it. So this is glucoamine because it used to be glucose. And this is galactoamine because this used to be galactose. So those are some of the common sugar modifications that we see inside the cell. And so here are some questions. So again, please pause and try this before you watch the answer here. But A, we have some bacteria produce what are called thiol sugars. A thiol sugar has a carbon-sulfur covalent bond, such as 2-deoxy-2-thiol-beta-D-glucose. Draw this structure. Well, the first thing we have to do is, well, I was going to say we would have to draw it in the ring form, but that's actually not the case. We can draw it in the linear form. So here's glucose. I'm sorry. I just looked. This is a beta. So the beta is telling us we have to be in the ring form. All right. So this is glucose. So the first thing we have to do is make the ring cyclize, make this linear cyclize. So if you remember, the way to do that is you take the alcohol group that's furthest away from your C double bond O, and that's going to do the attack right here. So 1, 2, 3, 4, 5, and this carbon ring is the 6 atom, so oxygen. So this is carbon 1, 2, 3, 4, 5, and then this carbon out here is going to be hanging out. Now it's beta. Beta means we're going to be pointing in the same direction. So we find our anomeric carbon, and the anomeric carbon is the carbon that was attacked. So that's this carbon. And since it's beta, the OH has to be pointing in the same direction as the CH2OH. And then our special modification is that it's a thiol at the 2 position. So deoxy, 2 position thiol, which means number 2 carbon, which is this one, no longer has oxygen, but it has that SH group. So thiol. And then just the rest of our OHs I'm going to draw here. And that is deoxy-2-thiol beta D-glucose. B, we're talking about another modification we didn't talk about in the previous slide. It's called a phosphorylation. So phosphate looks like this. So draw beta D fructose 6-phosphate. So again, we take our OH group furthest away from our C double bond O. Fructose is going to be a 5-member ring. So 1, 2, 3, 4. That one's going to be a 5. So let me draw that. 1, 2. There's our 5-membered ring. So this is our anomeric carbon. This is carbon 1. Well, sorry, that's bad numbering on my part. That should be carbon 1, 2, 3, 4, 5, 6. So carbon 1's always at the top. So technically that's carbon 2. And carbon 1, our CH2OH, is going to be here. Our H is pointing down here. Then let's see here. OH, 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 and also coming off of that though, that's not an OH, that's an H. So coming up here, so let's see, this is beta. So carbon 6 needs to be pointing in the same direction. So this is carbon 1, 2, 3, 4, 5, 6. So that's how the numbering scheme goes for the 5-member ring. You have 2 CH2OHs floating out into space there. And this should actually be an OH here, I believe. Yeah, because that was our anomeric carbon. So it has an extra OH. And so when you're looking here, if it's beta for the 5-member ring, you're looking to see, okay, am I pointing in the same direction? So let's actually check if I did beta fructose in the right direction. No, I did not. So I actually wrote alpha fructose because I was not paying attention. So let me just get my eraser out here. So for beta, in the 5-member ring, so it's a little tricky. Those of you who can hear that bird outside. So now I am beta, because remember, we're looking for that OH. We're looking for what used to be the carbonyl carbon. When that gets attacked, that becomes an OH. And we're asking what direction is this pointing? This is pointing up. Okay, if that is pointing up, then we see what direction is carbon 6 pointing. Well, that's pointing up as well. So they are both going in the same direction. So that's beta now. So I drew that right. And the last thing we have to change about this is beta D fructose 6 phosphate. So I need a phosphate on my 6 carbon. So right now I just drew regular beta fructose. So let's make it a phosphate. And that is a phosphate on carbon number 6. So beta D fructose 6 phosphate. So this exercise is really good at getting you to really understand how these rings cyclize, because you have to pay attention to the numbering scheme there. And like I failed to do when I was doing fructose, you have to realize, okay, am I doing alpha or beta? And originally I drew alpha, and then I realized my mistake. And so there is beta. So that's beta D fructose 6 phosphate. And for any of the questions that we go over today, if you don't quite understand something, always feel free to send me an email. Do not ever hesitate to do that. We can set up a meeting, or I can talk by email. Either one of those is fine. The last thing I believe for this PowerPoint is talking about the glycosidic bond. Now, what a glycosidic bond is, it's a bond that connects the anomeric carbon to anything else. That's all it is. It's if the anomeric carbon's bonded to something else. And these can be either O-linked or N-linked. What that means is that the bond coming from the anomeric carbon, it has to be connected to either an oxygen atom or a nitrogen atom. That's where the O-link or N-link is coming into play. Here, we're looking at nucleotides. This is the anomeric carbon. It's connected to a nitrogen that is called N-linked. Here, we have our anomeric carbon connected to an oxygen and CH3. So this is O-linked, right? It basically depends on what atom it's connected to. Now, glycosidic bonds are quite stable. You can kind of think of them like the peptide bond, that once they're formed, they're very hard to break. And if a glycosidic bond connects to sugars, they are no longer a monosaccharide, but rather they're called disaccharides. And you can go to trisaccharides up, or you can just call them a polysaccharide. So instead of like saying a pentasaccharide, you might just say it's a polysaccharide. And with this, we have some terminology that we have to cover. Reducing and non-reducing. So in your disaccharide, if both of your anomeric carbons are in a glycosidic bond, or if they're connected to each other, so I'll draw that. So I'm just going to draw very simple sugars. OH, OH. So here are my anomeric carbons because they're the carbon connected to two oxygens. And let's say I had a glycosidic bond. I have an O-link glycosidic bond connecting them. So oxygen, connect, connect. This is called non-reducing. And the naming scheme comes from a reagent. Don't worry about like exactly what non-reducing means. Just know if your glycosidic bonds your anomeric carbons are in a glycosidic bond with each other, that's a non-reducing sugar. However, if you have one free anomeric carbon, you then have a reducing sugar. So that's the difference between a reducing sugar and a non-reducing sugar. Are my anomeric carbons both in a bond, or is one free? So here are some exercises to help you understand that concept. So pause, figure these out, and then come back once you're done. So I'm going to dive right in here. First thing first, where's your anomeric carbon? So that's what I'll do. Circle the anomeric carbon. It's the carbon connected to both oxygens, as you just heard some dogs barking there. So sorry if you're wearing headphones. So anomeric carbon here, anomeric carbon here, right? So glycosidic bond connecting my anomeric carbons, non-reducing. B, let's look at anomeric carbons. So anomeric carbon right here, because it's connected to two oxygens, anomeric carbon right here. So this one, this anomeric carbon on the right does not have a glycosidic bond. So this is reducing. C, here, anomeric carbon, two oxygens. And we have to find our other anomeric carbon, which is right here, two oxygens. So that's kind of tricky with these 5-member rings. So remember, just look for the two oxygens. So this is non-reducing. D, well, anomeric carbon right here, there's no glycosidic bond. So any monomer would be reducing. So hopefully if you have that, if you have questions, let me know. I'll be happy to go over this. But we will go over the next PowerPoint in the next video here. So I'll see you there.